Hi friends, today we are going to see the past year NEET questions for the chapter Cell, the unit of life, and today in this video we will cover part 6. So already we have covered about 5 parts. So people, those who haven't gone through, kindly go through so that this will help you in the continuity with the chapter. So let's begin with the first question here today. Read it here. This question was asked in the year 2002. Which of the following occurs more than 1 and less than 5 in a chromosome? Okay, you're given 5 options here, right? You have chromatid, chromosome, centromere and telomere. Of course, telomere cannot be answered. So what is a chromosome first? We have to know what the answer is, right? When I say chromosome, you can describe them as thread-like structures. They occur as thread-like structures. Okay, thread-like structures in the nucleic acids, and they're present in the form of protein in the nucleus of the most living cells. What do they carry? If you see, they carry genetic information in the form of genes. So this is chromosome, and this is how you define. Now, what occurs more than 1 and less than 5? Okay, the correct answer to this question here is chromatid. I'll explain you why. See, a chromatid is one of the 2 identical strands of a DNA which actually helps in the making up of a chromosome that are joined at the centromere. So for this process, what happens here, if you see, it will undergo nuclear division. So when I say nuclear division, what are the 2 things that comes to your mind? Either it can be mitosis or it can be meiosis, right? We all agree with that. So the term is used till the centromeres remain in contact. That is, when the centromeres remain in contact, you will call that simply a nuclear division. But once they get separated during the anaphase, which happens during mitosis and meiosis, they are called as simply daughter chromosomes. Okay, so far understood? They are simply called as daughter chromosomes. Now, the tips of the chromatid, you call them as telomeres. Right. Now, these telomeres, what is a unique feature about this is, these telomeres, when they undergo continuous or repeated cell replication, these telomeres tend to dying. That is, they get their length shorter and shorter as the replication goes, and one part of time they vanish. So this actually dominates, or this actually determines the lifespan of the cell. So this chromatid is very important, and thereby it is understood this occurs more than 1 but less than 5 in a chromosome. Now you understand the correct option here is option A. Okay, so whatever I explained is to your right side, so you don't have to worry. The process is very simple. Okay, now let's move to the next question here. Again, this question was asked in the year 2002. This question is very easy. In fact, ribosomes are produced in dash. I think all of you by now would have guessed the answer. Yes, it is nucleolus. As we all know, Golgi bodies take part in the elaboration and secretion of the complex biochemicals, right? So mitochondria are the site of ATP synthesis, right? We all agree with that. So ribosomes, as you all know, it is the site of RNA and proteins. So ribosomes are produced in the nucleolus, and ribosomes do not have a proper cell membrane. The correct answer is option A, nucleolus. Okay, now let's move to the next question here. This question was asked again in the year 2002. Mitotic spindle is mainly composed of which protein? Again, this is an easy question. If you know what proteins are present in microtubules and microfilaments, I think you don't have to get worried about this particular answer, because mitotic spindle will obviously have what in them? They will have microtubules in them, right? Mitotic spindle will for sure have microtubules in it. So what is the protein that is present in microtubules now? It is tubulin. Therefore, the correct answer to this question is option C, tubulin. Okay, to your right side you have a small picture telling you something about microtubules. They are protofilaments, they line up side by side, they are dimers, they disassembly faster, they are used to construct a cilia and flagella. So these are some of the functions of the microtubules. See, these kind of questions are easy when you go in depth with the lesson and study, right? In order to understand this question, you need to know what is a mitotic spindle composed of. As we all know, it is composed of microtubules, and therefore the protein present in microtubules, it's tubulin. Therefore the answer to this question will be easy for you. So the correct answer to this question is option C. Next question here. This question was asked in the year 2001. Okay. So here comes the question. Microtubules are absent in dash. We know it is obviously present in spindle fibers. We know it is useful and it is present in flagella. For sure it is present in fibers, and for sure it is present in flagella. Now, you only have to decide between these two, mitochondria and centriole. But microtubules are also present in centriole. Okay. Now, microtubules, as we already saw in the earlier slide, microtubules form dimers. Right. Microtubules are formed and they are dimers. Right. Microtubules are present in centriole, flagella, spindle fibers, but they are not present in mitochondria. Mitochondria are a site of ATP synthesis. Okay. Therefore, the correct answer to this question is option A, mitochondria. Okay. See, these questions seem to be very easy only if you had gone through the earlier parts, because in the earlier parts I've already explained in depth about microtubules, mitochondria and these things. So these questions seem to be easy if you had already gone through the earlier parts. Next question. Now this question again asked in the year 2001. Element necessary for middle lamella is dash. Okay. Now what do you mean by middle lamella? Where is it present actually? Any idea? See, when we talk about the plasma membrane, or when we talk about the cell membrane, whatever you can call it as, okay, you have something called middle lamella, then you have something called outer, inner, you have it like that. So when we talk about the middle lamella, it is a layer which cements the cell walls of the two adjoining plants together. So already we have discussed in the part 5 something called cementing, or you can simply call it as a glue. Right. So here the element necessary for the middle lamella to have that property, the correct answer here is calcium. I think this part was already explained to you in the earlier part as well. The middle lamella is a layer which cements the cell walls of the two adjoining plant cells together. It is the first form layer which is deposited at the time of cytokinesis. The major important constituent required or an element that is required for the middle lamella is calcium, because it is composed of it. Okay. Zinc is an activator of enzymes like carbonic anhydrase. So you have some important since calcium is the answer, what is the property of zinc, potassium and copper they have given? Zinc, if you see, it is the activator of enzymes like carbonic anhydrase. What does copper do? It helps in electron transport. And potassium, it maintains the membrane permeability. What do you mean by membrane permeability? Selectiveness. Right? Selectiveness. So the correct answer to this question is option A, calcium. So here to your left you can see a diagram which has the middle lamella, primary cell wall, the plasma membrane, and middle lamella is highly necessary of this calcium. Okay, otherwise it cannot function properly. Next question here. This question was asked in the year 2000. Okay, the cell organelle involved in the glycosylation of protein is dash. Okay, so this question is again easy for you. When we talk about glycosylation, glycolipids, Okay, all this process happens in the reticulum. That is, we already know they are classified as smooth endoplasmic reticulum and rough endoplasmic reticulum. These two reticulums have a separate property and function. While smooth will produce or will synthesize lipids, and this will synthesize protein. So when we talk about glycolization, the correct answer goes to endoplasmic reticulum. And now, what is glycolization? Glycolization is a process or a result of addition of saccharides to the proteins. So you're adding sugar to protein and lipid. It's glycolization of protein. You're adding two things to sugar. Okay, so this is called as glycolization. This process is one of the four principal co-translational and post-translational modification step. So you have to understand how important is this step, right? So here, if you see, this process of glycolization will take place, or it involves the endoplasmic reticulum, which actually depends on the smooth and the rough endoplasmic reticulum. Okay, because smooth will synthesize lipid while the rough will synthesize protein. That is why endoplasmic reticulum plays a vital role here. The correct answer to this question is option C, endoplasmic reticulum. Okay, next question here. This question was asked in the year 2000. Okay, lysosomes have a high content of dash. What are lysosomes? Any idea? Where do you find it, or what is its activity? See, when I say lysosomes, they are a membrane bound cell organelle. We all know that, right? Lysosome is a membrane bound organelle. So does it have a cell wall? Does it have a cell wall? So what does it contain actually? See, this one contains digestive enzymes. Lysosomes are involved in various cell processes in a human cell or in a living organism. What is the actual function? If you see, it helps in breaking down molecules. Okay, they break down excess or worn out cell parts. They may be used to destroy invading viruses, especially invading viruses and bacteria. Okay, therefore this lysosome have high content of option A, hydrolytic enzymes. Okay, they are mostly, if you see, these lysosomes, they are single membrane bound organelle. I already asked you during this solution part, do they contain a membrane or not? They have a membrane, but it's a single membrane. Okay, they are rich in acid hydrolases. So that is why lysosomes have a high content of hydrolytic enzymes. Therefore, the correct answer to this question is option A, hydrolytic enzymes. Next question here. This question was asked in the year 1999. Okay, some of the enzymes which are associated in converting the fats into carbohydrates are present in dash. Okay, this question is also easy for you. So if you clearly understand this, which or some of the enzymes which are actually associated with converting fats into carbohydrates. Okay, you're given four options here: liposomes, microsomas, and Golgi bodies. Okay, and you also have glyoxysomes. So we'll discuss one by one. Okay, as we all know, Golgi bodies, they are cytoplasmic organelle which take part in the elaboration and secretion of the complex biomolecules. Now, when we talk about conversion, that is fats into carbohydrates, this is carried out in glyoxysomes. Okay. Now, how about these liposomes and microsomes? What about these two features? Now, microsomes are the small single membrane cell organelles that absorb oxygen. Okay, so what do? They absorb oxygen. Okay, and they perform direct oxidation method. I mean, they carry out oxidation of substrates. Now, if you talk about liposomes, they are also involved in some kind of this activity, but here if you see, it involves mainly lipids. Okay, So what about glyoxysomes? Glyoxysomes contain enzymes for beta oxidation of fatty acids. That is why the conversion takes place in glyoxysomes. Okay. Now you understand why is it glyoxysomes. Therefore, the correct answer to this question is option D, glyoxysomes. Okay. Next question. Now this question was asked in the year 1999. Let's read it here. Which one of the following organ has a single membrane? Very easy question to answer, I think. We already discussed in the earlier parts the organelles which have single membrane, double membrane and no membrane. So we all know what the answer is. Of course, nucleus has a membrane, cell wall has a membrane, mitochondria has a membrane. Out of all the four options, if you see, cell wall, then mitochondria and nucleus, these three are actually double layered membrane or double wall membrane. They are all double. So the leftover option is spirosomes. The correct answer is option D. Nucleus is enclosed. Nucleus, cell wall, mitochondria, they all have a double layered envelope. When you talk about spirosomes, they are the small spherical single membrane bound cell organelles. What is the activity? If you see, the activity is to synthesize and store fat. Okay, their main activity is to synthesize and store fat. Mitochondria is also surrounded by double layered membrane which has an outer membrane and an inner membrane. Cell wall, you know, obviously it has a membrane around it. Therefore, the correct answer to this question is option D, spirosomes. Okay, see these questions are very easy to answer when you distinguish which and all, if you can keep in your mind which is double layered, which is single layered, when which has no membrane. Okay, next question here. This question was asked in the year 1999. Very easy question, in fact. Again, the proteins are synthesized in dash. The proteins are synthesized in dash. It's an easy question here. We already know mitochondria are responsible for the synthesis of ATP. Right? They are responsible for the synthesis of ATP. So likewise, everything has an activity here. So Golgi complex, if you know, it is its activity is to its activity is in the elaboration and secretion of some biomolecules or biochemicals, whatever you can call it as. Centrosomes, they are involved in centrioles part, right? The distinctly staining region of the cytoplasm surrounding the pair of centrioles together will constitute to form the centrosome. Therefore, the correct answer to this question is option A, ribosomes. We all know ribosomes is the region where RNA and protein synthesis take place. Therefore, the correct answer to this question is option A, ribosomes. I already said mitochondria is involved in ATP synthesis, Golgi bodies in biochemicals, while centrosomes, they are a part of the centrioles. They are dependent on the centrioles. All the centrioles together with the other centrioles will form together the centrosome part. Therefore, the correct answer to this question is option A, ribosome. Next question here. This question was asked in the year 1997. Okay. Genes located on the mitochondrial DNA. Right? The genes that are located in mitochondrial DNA. Fine. So we'll go one option by option. Generally show maternal inheritance. Option B, are always inherited from the male parent. No, that we cannot agree. C, show biparental inheritance like the nuclear chains. That is also wrong. And the last option is again where they talk about no inheritance. Okay. C, genes that are located on the mitochondrial DNA, okay, they are inherited only through which one? They are inherited only through the cytoplasm. Very important, right? Through the cytoplasm. The zygote receives the cytoplasm from which one will it receive? It will receive from the female gamete. Right? It will receive from the female gamete. Hence, since it receives from the female gamete, it is very well understood it receives the information from the maternal inheritance. Therefore, the correct answer to this question is option A, generally show maternal inheritance. Okay, now you understand why is it maternal inheritance. Okay, next question here. This question was asked in the year 1995. Okay, fine, let's read it here. What's the question here? The function of the rough endoplasmic reticulum. This is a very easy question. By now you would have guessed the answer. Yes, the rough endoplasmic reticulum is involved in the protein synthesis. As we all know, endoplasmic reticulum has a smooth and a rough part. Okay, as we all know, smooth is responsible for lipid synthesis. Now rough is responsible for protein synthesis. Therefore the correct answer to this question is option C, protein synthesis. To your right side you can see a diagram which shows the rough and the smooth part. Along with that you also have nucleus, nuclear pore, then you have the ribosomes, all that is there. Okay, so this is responsible for lipid and this is responsible for protein. Okay, easy question in fact. So let's move to the next question here. This question was asked again in the year 1995. The point at which the polythene chromosomes appear to be attached together is called as dash. Okay, so what do you mean by polythene chromosomes? Right, you need to know what is polythene chromosome. We just now saw what is a chromosome. What is chromosome? They are called as thread-like structures which are present in the cytoplasm. Likewise, when we say polythene chromosome, poly means many, right? We all agree with that. Now what do you mean by polythene chromosome? They are chromosomes which are large. As I already said, poly means many, so they will be large or huge, which have thousands of DNA strands will be present in it. Okay, they provide a high level of function in certain tissues such as salivary glands. Polythene chromosomes was first reported in the by any idea? Already we have seen this answer here, by Balbiani in 1881. Okay, now coming back to the question. Right, we are coming back to the question here. So the question is about what it seems to be what appears to be attached together. What do you call that as a polythene chromosome? Now here the correct answer goes to chromocenter. So granules of the condensed chromatin, which you call it as hetero means different, so heterochromatin, they are found scattered at the interface nuclei. We all know that. At chromocenter, the polythene chromosomes appear attached. So which part? Chromocenter. Therefore the correct answer to this question is chromocenter. So what is a polythene chromosome? They are large chromosomes. So what the main activity of this polythene chromosomes are seen in salivary glands. Okay, salivary glands. So option D is the right answer for this. Next question here was again asked in the year 1995. The desmosomes. What is it? Desmosomes are concerned with dash. Okay, so you're given 4 options here. Cytolysis. What do you mean by lysis? Breaking or killing, whatever. So cytolysis means cell breaking, cell division, or it is during or is it concerned with cell adherence or cellular excretion. So what is a desmosomes? See, desmosomes are actually they are the how can I describe is that they are the cementing substances. Okay, you can call them as cementing substances. Okay, so I've already given you the clue. When I say cementing, it means it is sticking, or you can call it as adhering. Okay, so this cementing effect, if you see, will help in the adherence of each other and keep the latter tightly packed as they prevent the leakage of the fluid from the interface. Okay, so desmosomes are concerned with cell adherence. They prevent the leakage, which is very, very important. Okay, Therefore, the correct answer to this question is option C, cell adherence. Okay, next question here. It's very easy question asked in the year 1994. Inner membrane convolutions of mitochondrion are known as dash. Easy question only. So what is it actually? Any idea? What you mean by convolutions? Any idea? See, you're given 4 options here. What are the options given here? Look at the options. Lamella, we already saw what they are. Grana, you know it is a part of the chloroplast. Okay, so what do you mean by convolutions? Okay, convolutions. So if you see, this convolution can be described as a mathematical operation of 2 functions. In mathematics, if you see, they will express it in the form of mathematical operation, and it will have 2 functions, it seems. People, those who know well, Max well known, they will represent as f or g. Okay, when we talk about this particular sequence here, when you relate it to Max, it will be easy for you to understand, so I have related in such a way. So they will have 2, the operation will be in the form of 2 functions. Okay, so the term convolutions or convolution, they refer to both result, that is 2 functions. Okay, so therefore the inner membrane, okay, the inner membrane, what do they contain? If you see, they contain cristae. The inner membrane of mitochondria is folded or unfolded? It will be unfolded. Okay, it will be unfolded to form involutions called as cristae, and they are meant for increasing the active area of the inner membrane. Therefore, the correct answer to this question is cristae. Now, what is cristae, people? It's very important that you need to know what is cristae, right? Cristae, if you see, can be defined as the partial partitions of the mitochondria. How can you describe it as? Partial partitions. See, at one part they might be folded, at one point they might not be folded also. So, cristae are the partial partitions that are present in the mitochondria, formed by the infolding of the inner membrane. Very important point. See, without knowing what is cristae, just blankly learning will also not explain. Therefore, it is very important that whenever you see questions like this, you learn along with the options. Okay, just don't blankly read the answers. So, cristae are partial partitions that are present in the mitochondrion or formed in the mitochondrion, which is called as the infoldings, or which is formed by the infoldings of the inner membrane. Therefore, the correct answer to this question is option D, cristae. Okay, so with this we complete part 6, cell, the unit of life. We will meet in the next part. Thank you.